Very good afternoon to everyone. Today we will study about the basic and most important cell organelle of a cell, and that is nucleus. It's the controlling power of a cell and it's also called as the controlling center. It has the genetic material of a cell, and the genetic material of a cell is very important to make it living. The nucleus was discovered in 1831 by Robert Brown, and he has described nucleus as the cell organelle of a cell. Later, the Fleming was another scientist who has stained the material of the nucleus with a basic dye, and he said it as chromatin. Later, we know that eukaryotes may be uninucleated, means they have only one nucleus, or they may be multinucleated, or they may have one or more nucleus. But the special point is that there are few cells which doesn't have nucleus, and it includes RBC, the erythrocytes of mammalian cell. The RBC initially have nucleus, but after maturation they lack the nucleus. Second example is the sieve tube or the sieve tube cells of vascular plants, and these cells also lack nucleus. One more special example of a nucleus include paramecium. Paramecium is a very special organism, and it has two nuclei. First is the macronucleus and the second is the micronucleus. Micronucleus is involved in the sexual reproduction, while macronucleus is involved in different vegetative functioning, and the first is the feeding, second is the waste removal in the paramecium, and third is the water balancing in paramecium. Thus, the two nucleus of a paramecium, macro and micro, has different and diverse functioning in the same organism. Now, coming to the interphase nucleus of a non-dividing cell. When a cell is not dividing, it's in the interphase, it's having four parts. The first part is the nuclear matrix, the matrix that has all the contents of a cell, and secondly, it has extended and elaborate nucleoprotein called as chromatin. Thirdly, it has one or more spherical structures, and it is called as nucleoli, or singularly it is called as nucleolus. Thirdly, all the content, nuclear matrix, chromatin and nucleoli, are covered by an envelope, that is a double membranous structure, and it is called as the nuclear envelope. Thus, the nucleus of an interphase cell has four parts: matrix, chromatin, nucleoli and envelope or nuclear envelope. Now, coming to the basic structure of the nuclear envelope and nucleus. A cell has a nucleus with a nuclear envelope. The nuclear envelope of a cell has two coverings, that is a double membrane bound structure. It has an outer nuclear envelope and an inner nuclear envelope, but the two nuclear envelope together has a space that is called perinuclear space, and this space has a gap of about 10 to 15 nanometer. This space was discovered by the discovery of electron microscopy. The outer nuclear envelope is in continuation to the rough endoplasmic reticulum that has the ribosomes over its surface, and it is very actively involved in protein synthesis. At some points, the two nuclear envelopes are fused together and they form a nuclear pore. This nuclear pore is a very complex structure, and about 3000 to 4000 nuclear envelopes are present in a cell. The fusion of two nuclear envelopes form the nuclear pore, and this is having a cylindrical structure with an octagonal symmetry. The symmetry has 8 molecules together, and it has about 30 different proteins. This nuclear pore is very important, and it has been involved in the bidirectional transport of RNA and protein from nucleus to the cytoplasm and then back to the cytoplasm to nucleus. With this, a nucleus has chromatin material and nucleolus, that is a very specific special and spherical structure. It is a membrane-less structure and it is in continuation to the nuclear matrix. This nucleus is very important and it is involved in ribosomal RNA synthesis. If a cell has numerous and a larger nucleus, means that cell is very active and involved in protein synthesis. Now, after studying the non-dividing cell, we will study about the dividing cell. A dividing cell or a cell after interphase is showing the condensed chromosome structure, that is the condensed chromatin fiber. This condensed chromatin is called as the chromosome and it has 4 parts: DNA, histone protein, that is a basic protein, non-histone proteins, and RNA. If we stretch this chromatin fiber, it makes a 2-meter long DNA fiber, and this includes the whole chromosome set or 46 chromosome or 23 pair chromosomes of a cell. Now, the most important part of a nucleus, that is chromosome, that is the condensed chromatin fiber. The chromosome has this structure and it has 2 homologous chromatids known as chromatid 1 and chromatid 2. The 2 sister chromatids are joined together and they have a small primary constriction that is called as centromere. This centromere is enveloped by a disc-like structure that is called as kinetochore core, while some chromosome has secondary constriction that is present at some constant positions and give a small fragmentous structure to these, and it is known as satellite or secondary constriction. On the basis of presence of centromere, chromosomes can be divided into 4 types, that is metacentric, submetacentric, acrocentric and telocentric. The type of chromosome include first metacentric. The 2 chromatid is known as P and Q regions. If the P and Q regions have equal length and centromere is present at the center of this, is known as metacentric. In submetacentric chromosome, the centromere is towards the P chromatid and the 2 chromatids P and Q have a difference in their length, and this type of chromosome is known as submetacentric. While the third type is acrocentric chromosome, where the length of P is Q is variable and the centromere is closer to one of the end of the chromosome, and this is called acrocentric, means close to one end. The fourth type is telocentric chromosome, where the centromere is present at the edge or end of the chromosome.